Now let's talk a little bit more about flow cytometry. This is an extremely powerful tool which is used in many applications in modern times. And as the name implies, we have flow, cyto, and metry. So metry is measurement, cyto is cells, and flow means a fluid suspension. So basically, flow cytometry is measurement of cell properties such as shape, size, expression of surface markers or immunophenotyping, and even expression of intracellular signaling molecules in a fluid suspension of the cells. This is achieved by passing a flow of cells in a single line labeled with fluorescent antibodies against markers of interest into a system of lasers and detectors that transmit fluorescent emitting to a computer, which allows data analysis. And therefore, the components of a flow cytometer are a system of fluidics, optics, and electronics. So to determine a cell population carrying a specific marker by flow cytometry, you can obtain a sample of cells from the patient, incubate the cells with the fluorescent antibodies against markers of interest, run the cells through the flow cytometer, and then analyze on the computer using a specialized software in different cell populations. So let's see now how flow cytometry can be used to count and analyze the size, shape, and properties of individual cells within a heterogeneous population of cells. Flow cytometry data is extremely quantitative and can be analyzed in depth by specific software programs. Once a suspension of individual cells is prepared for flow cytometry analysis, the prepared sample is placed on the flow cytometer. Now, how does a flow cytometer analyze the size and shape and properties of individual cells? It contains several key components, including the sample, fluidics that move the sample into the flow cytometer, lasers, optics which gather the light, detectors to sense the light, and a computer system to output the data in a form that can be analyzed by the flow cytometer. Once the sample is placed on the machine, it will be sucked up, it will be mixed into a stream of saline solution into the cytometer, and the cells pass through the chamber in a single line before they pass through the laser. Each cell within the sample then moves across the laser beam, allowing each cell within the sample to be individually analyzed. Let's take an example. In a patient suspected to have a disease associated with a disturbance in regulatory T helper cells, which I hope you remember are CD25 positive, CD4 positive T cells. So you take a sample of his lymphocytes and you stain the cells with two fluorescent antibodies. An anti-CD4 antibody, which emits green fluorescence, and an anti-CD25 antibody, which emits pink fluorescence, just as an example. So what you can see over here is the result that you see on the actual computer. Based on these results, this is a dot plot, what proportion of the patient's cells are CD4 positive? 60% are, and I'm talking about all the upper quadrant population of cells, anything that is expressing high levels of CD4. Now, what proportion are CD4 negative? 40% are CD4 negative, everything that is low on the green fluorescence. What proportion are CD4 positive, CD25 positive T cells? That would be 40%, the upper right quadrant, so it is expressing high levels of both CD4 green fluorescence as well as CD25 pink fluorescence.